In this video, we will discuss about this HT-VAC or HT vaccines efficacies against the clinically relevant mice system. So what do you mean by clinically relevant mice system? Is that they develop or they use mice system where they inject the tumors at two different sites. At distant site, they have injected a less tumor cells, that is around 0.1 million, and at primary tumor sites, they have injected around 1 million cells. And both the injections are subcutaneous, so that the point of injection remains intact. And in this case, the primary tumors means that at this site they have introduced the virus to express this hepatitis B antigens on the tumor cells. First they check whether this system, where first they introduce the mRNA with hepatitis B mRNA, hepatitis B surface antigen mRNA, so that the immune systems will get primed or have generate the antibodies against the hepatitis B surface antigens. And thereafter they introduce the tumors at two different sites, and then they inject this virus that can express hepatitis B on the tumors. And after this 10th day of the tumor injections, they introduced their vaccines along with this anti-PD-1, and they use the anti-PD-1 because at the secondary tumor sites there will be tumor microenvironment that can exhaust the activated T cells. In order to prevent the exhaustion of the activated T cells, this anti-PD-1 is used. So the results shows for the primary tumor sites is that the results shown for the primary tumor site is that the tumor suppressions is maximum when they use HT vaccines or HT vaccines plus anti-PD-1. And these things they have shown individually in the panel, first row of the panel D also, where HT-VAC and HT-VAC plus anti-PD-1 shows maximum tumor suppressions. And for the distant tumors, they are able to see tumor suppressions maximum for both the cases. However, this anti-PD-1 outperforms anti-PD-1 plus HT-VAC outperforms the HT-VAC alone. And these things are also shown individually in the panel D, where we can see that the HT-VAC plus anti-PD-1 shows high performance or more tumor suppressions compared to the case where HT-VAC alone is used. So this indicates that their vaccines is able to generate endogenous T response against the distant tumors also, and its performance can be increased by combining it with checkpoint inhibitors therapies. And after that, they checked it for another relevant conditions where the vaccines is usually injected when the tumors are detected. In that case, they first inject the tumors, and after 4 days they inject this mRNA H to generate the antibodies against this hepatitis B, and at 18 days, 20 days, 22 days they introduce the virus. And then they check whether in this case also the performance of the HT-VAC is better than the controls or not. And they are able to see that when they use the HT-VAC, they are able to see maximum tumor suppressions compared to the controls. So here also the results are consistent with the performance of the HT-VAC outperforms all other cases. Thereafter, finally they checked it for the highly metastatic liver cancer cell lines, that is H22 luciferase cell lines. So here the model system is same, that is they first introduce the mRNA of hepatitis B, and then they use this inject this H22 cell lines, and after 5, 7 and 9 days they introduce this they inject this virus that can express hepatitis B surface antigens of the tumors. And in this case also they are able to see inhibited tumor suppression inhibited they are able to see that the HT-VAC is able to inhibit the tumor growth compared to the control case. And when they check the immunofluorescence image, it also shows the same things, that in the case where HT-VAC is used, the tumor progression is slower or delayed compared to the control case. And when they check the survival rate, here also they find that survival rate in the case of this HT-VAC outperforms than the control. That is, survival rate when HT-VAC is used is increased. The survival rate increased from 0% to 80% when HT-VAC is used. So all these results along with the previous results shows or underscore the strong anti-tumor efficacies of this HT-VAC that they have developed. So I hope you understood their results and the points that they want to make. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.